Well, thank you, Linda, for that introduction. We've got a terrific group here, and I don't want to take a lot of their time, but I think all of us felt that it was a good idea to take some time this morning to lay out where we're at with this whole question of what some people call personalized medicine, others have called precision medicine. It's a very simple idea, and it actually came up in the last question for Adam, and that is, if you begin doing clinical trials, you find out very quickly that no matter what your intervention is in this whole area of mental disorders and addiction, you're looking at a very mixed bag. It's a little bit like doing a clinical trial of a new antibiotic and taking in everybody who has a fever. And it's not surprising that the antibiotic only helps about half of them, because half of them are likely to have a viral infection. And yet that's what we do. And it's part of the reason why we've had so much difficulty in this field in either demonstrating the efficacy of new interventions or trying to understand how any of them work. It's because we have a fundamental problem around diagnosis. And Linda's comment about what NIMH has been trying to do, one of our major projects has been this research domain criteria project, which just says making diagnosis entirely based on symptoms is just not going to get us to where we want to be. It would be like saying that everybody who has a fever has the same disorder, or everybody who has chest pain has the same disorder. We don't do that in the rest of medicine, and we have to figure out a way to get past that when we are talking about people with these brain disorders. So how do we do that? Well, you're going to hear three approaches to beginning to take this on from people who have thought very, very carefully about this, and I understand at least two of you have PowerPoint presentations, so we'll make sure that you have a chance to go through some of those slides, and then we'd love to have some time for discussion. We'll start with Dr. Evian Gordon, who's the executive chairman of My Brain Solutions. He established what has been called an integrative neuroscience approach, grounded in the use of standardized methods across multiple types of brain data, and you're going to hear a bit about that for depression. He founded the Brain Resource Company that created the first integrative international database on the human brain. The database is the asset which shapes the development of mybrainsolutions.com for brain health, used in wellness programs in over 50 well-known companies and Fortune 500 company employees, and over 50 recovery programs and addiction clinics. The first brain test using this methodology for personalized treatment prediction in depression has been published and submitted to the FDA for potential widespread use in clinical practice. Evian, would you prefer to use the podium? Great. So
I can use the slides.
All right. Well, there is a clicker here, and that'll allow you to advance.
Thank you. Thank you, Tom. And it's a privilege to be here again. And thank you for the invitation, Linda and team. And I want to share with you the lessons that we've learned as an international consortium of scientists, about 250 scientists from 28 countries, who, together with Steve Koslow from the Human Brain Project, the inaugural head, I believe he was, at NIMH, Tom, I believe he was one of your employees, to share some of the lessons of big data. And our focus is what works in terms of biomarkers. What is real about treatment prediction for any psychiatric disorder? I happen to going to briefly show you a few snapshots of depression and addiction. But we started, I'm going to give you an exemplar of a study that John Rush, who was, of course, the PI of STAR-D, who Tom funded, and his team, John's the chairman of our scientific publication committee of this particular study, iSpot, international study to predict treatment response in depression. And the reason I choose that, there are 22 publications. If you want to see them, there's the address. And there's also, this is a for-profit group that ran this study. But there's also a not-for-profit foundation where it's actually 350 scientists from 28 countries called BrainNet.net that have published 400 papers on biomarkers using the same methodology, the same data in over 10 different psychiatric disorders. And I suppose I just want to share with you what have been some very, very challenging lessons. But my takeaway, I always try and start with the end in mind, is that the use of biomarkers in psychiatry is not a future event. These 22 publications, in my view, and I'll leave you to judge for yourself if you look at the papers, are proof points that if you run large enough studies with the right statistical power, with well enough clear questions about remission, many of the measures that are currently standardized and in use can predict treatment response. Now, it remains to be seen the extent to which these studies are replicated, and that, of course, there's nothing in science that is relevant unless it's replicated. But it is not a future event. My prediction is that within the next 2 to 3 years, whether it's these treatments that I'm about to show you, or whether it's the gaming that Adam so elegantly represented, or an example that I'll end off with an addiction that we use, any intervention, the takeaway message for us that was a hard-learned lesson is that this is very difficult to do and get a result that is replicated with small numbers. So the study essentially was funded by a European biotech. Unfortunately, it's just a $20 million study, so that's the order of magnitude you're talking about for each study. And if this happens to be a study, and by the way, Tom Insel was very integral in helping us speak to the study people who we benefited from. They were magnanimous, John Rush in particular. And this was part of the learning of drawing upon the previous experience that exists. And this particular study required literally 1,700 subjects, 1,000 in the training set. And may I also say that the FDA device team were very helpful to go to right at the beginning of the study and tell us how to design it. Get a training set, get all the data you can, optimize your algorithms, do cross-validation that gives you the best chance of predicting whether it'll replicate, and then run a replication-independent sample. This was tough stuff. It took 7 years, 30 sites, mainly around the United States, actually 30 sites, not 20, and around in Europe and Australia, and the training cohort, and then the replication cohort of pretty much everything that moves in the brain. That was the other thing, to see what works. And I'm just going to give you a snapshot. It'll literally take me less than 3 minutes now to show you some of the publications as to the sorts of markers that can be used, potentially, we'll see what replicates, to predict treatment response of who should and should not be on the 3 most commonly used antidepressants in psychiatry that were in the previous slide. Escitalopram, sertraline, and venlafaxine. So in cognition, for example, you can predict in the training set that sertraline, you can predict who won't respond to sertraline. It's pretty high accuracy. And as John Rush says, for a clinician, sometimes to know what doesn't work is as important as to know what does work. And I suppose I also want to express my gratitude to Henry Harbin, who has also provided the constant context, and I believe that the FDA have this context, frankly, from what I've seen, that perfection is the enemy of good. Nothing happens in one big step to suddenly develop the first test in the history of psychiatry, which still remains, as we know, the only discipline without tests that predict treatment response. It's what is better than the current level of evidence, current clinical practice. And you only have to be 10% better than that with some objectivity to be meaningful. So thank you, Henry, wherever you are. You've been a constant source of inspiration and kept many, many analysts going in the 40,000 analyses that were done just for this study alone. The second example is genetics. Alan Schatzberger has been spending 20 years looking at this one gene, ACBC. And for those of you who don't know, it's a gene that determines, if you've got a variant, whether you pump any brain drug through your blood-brain barrier into your brain, or more likely to pump it out of your brain, and this has massive effects for dosing. So some people who are oversensitive to any drug, if you don't know your ABCB1 status, it kind of has some very serious implications for your dosing. And Alan was kind enough, and as you all probably know, he's the ex-president of the American Psychiatric Association, to guide a team through just to look at that gene, and then using the cognitive battery that I previously showed you. It's just an online standardized cognitive battery, probably the most validated cognitive battery, that also has the same cohort who have MRIs, DTRs, fMRIs, genetics in the same subjects that I'm aware of. But this single gene could help predict escitalopram and sertraline, both positive and negative predictive values. So, of course, we're now looking at the other 700 SNPs or so, but that's hard to imagine, even with the new neurochip, which is, we're about standardization, of course, which I failed to mention. That's our kind of, if we do have one innovative piece, in addition to running studies on scale, it's standardized, everything. Every cognitive piece is standardized. So anybody around the world who uses any piece of our methodology, it's all standardized. So we really then benefit from the power of numbers. So when people run genetic tests and they're using hundreds, let alone hundreds of thousands of SNPs, it's incredibly unlikely that anything is going to replicate, and the proof is in the pudding. But if you select your genes and your SNPs based on previous evidence such as this, we're currently looking at 600, but have selected out 15 that have had previous evidence of some, at least snippet, of clinical relevance. And EEG's wonderful technology. And we've got, this is just an exemplar finding of asymmetry, and we're all familiar with the notion, the simplistic notion, frankly, that your left hemisphere may be responsible for positive emotions and your right hemisphere associated in some way with negative. But the fact of the matter is that this a simple EEG asymmetry measure can predict escitalopram and sertraline response, and a theta EEG activity with Loretta analysis, including the anterior cingulate, can help predict venlafaxine. Now, again, replication. We're doing it at the moment, but these were very promising first signs, and each modality has different teams around the world using the key opinion leaders in that sector. And last but not least, of course, is MRIs and DTIs and fMRI. This just happens to be an MRI example where left frontal, middle frontal, and right angular gyrus volume can help predict response to all three of the medications or not. I believe this is the first study in which there's been a publication in the same cohort for each of those domains of biomarkers. And I think what's important about that, and our general database, which now has hundreds of thousands of people, is we can look now at the correlation, the cross-correlation of the biomarkers. So you run a cognitive, simple study online, and then you can then see what's the correlation to the genetics, to the MRI, to the fMRI, to the DTI. And that augurs well for testing out the sort of neurogaming type approaches that Adam was talking about, because it's very hard to run studies on this scale and expense. I'll end off with addiction. We have 100 addiction clinics around the country. There are 14,000 addiction clinics, as you probably know, that do MyBrain Solutions, which just is a set of a simple cognitive assessment, the same one, all standardized, and some training tools like a relaxation room for mindfulness meditation, plus some breathing bars, which turn out to be quite important, and a thought tamer, which is just cognitive behavior therapy gamified. And with 3 hours of training over 30 days, the first 2,000 patients, this is data from the first 2,000 patients, improved their symptoms significantly, decreased their symptom level significantly, and there was a smaller number of dropout rates in these, in a significant number of these clinics. So just an exemplar again that it can also be done without $20 million studies, but you need scale, you need numbers to really see the statistical power. And the last is just a snapshot. John Rush looked at a marker called negativity bias, which you're all familiar with, literally from the earliest days of depression, that people who magnify threat, 100% of patients across all disorders in our database, 10 disorders, who had depression had a negativity bias marker. In anxiety disorders, something like 85% have some disturbance in heart rate variability, which, as you know, is very linked to your hypothalamo-pituitary-adrenal axis. In ADHD, theta remains still a very interesting marker, and I know you'll probably all know the FDA has accepted theta beta ratio as its first biomarker diagnostic. And PTSD, non-conscious processing, that's with non-conscious stimuli presented so fast that you can't actually see them, of faces, using Rubin-Ger's faces, activating the amygdala was a really interesting biomarker. In people with schizophrenia, over 20 papers showing timing dysmetria. Nancy Andreasen calling it cognitive dysmetria was a very foresightful, either very clever, intuitive guess or just sheer brilliance, I'm not sure which, but she was right on the money. There's timing disorders across anything we measure in the 20 studies in people with schizophrenia, and of course Alzheimer's speaks for itself, and I will in that little snapshot there. Thanks very much.
Thank you. I think we'll hold questions till the end, and then we'll have a whole panel discussion around this. Let me introduce our next speaker. George Carpenter is CEO of CNS Response Incorporated, leading the commercialization of the company's patented peer technology for psychotropic medication management. CNS Response is the first biomarker solution for providers in behavioral medicine, a results-oriented biomedical executive with a passion for leading high-growth and turnaround companies. George is focused to bringing new technology and business processes to underserved markets has been really a MO for a long time. He's done this in a number of different areas. So, George, great to have you here and looking forward to your remarks.
I'm very grateful, Tom, and Tom is one of my heroes for making this an issue that I think we're all tuned into. And my panel members, I'll say right at the outset, what we're doing is very complementary with what my other panel members are. But there is one thing that makes us unique, which is I think we're the only company that has both been featured in Oprah Magazine and Soldier of Fortune. And we've been working with the military since 2010. And you know, the bogey that we're trying to beat is it took 200 years for the Royal Navy to adopt citrus fruits on board after the good epidemiological work was done showing that it would cure scurvy. We're going to beat that number. And you know, the military is a great test bed for this kind of science because they truly are risk-seeking, interested in pushing the science, and they have large numbers. So let me just kind of go to the issue of what we do. We help predict medication response using some of the things that Tom and Evian talked about. Quantitative EEG features, the 5,000 variables that are spit out of a quantitative EEG, map pretty well to certain characteristics of medication response. EEG is one of the least expensive neuroimaging units that you can use, and it's reimbursed by most insurance carriers. When you combine that with a large outcome registry, we've done 12,000 peer reports. We have 10,000 unique patients in the database and 38,000 endpoints. We've been able to narrow down medication response. You know, literally, how great would it be if your doctor had a report that could just tell him, you know, here are the drugs in the red, here are the drugs in the blue. We use red, white, and blue because it's the military. Stay out of the red. Would that be helpful? Well, what we found is in every study we've done, there's been a 2 to 3 times improvement in medication effectiveness. And as we're beginning to do and study endpoints now, reduction in risks, including suicidality. So the outcome registry, this low-tech idea, which is now really gaining currency, ends up being the secret sauce. And by putting it in the cloud, we're able to do it everywhere, all the time, night or day. So the network really is in the computer. And as I said, you know, we're very transparent in open science. We believe that a doctor gets a report, they ought to know where it came from. They ought to literally be able to see every dot on the map that created that report and see the type 1 and type 2 error. Really, what is the true positive, true negative in this report? But the coolest thing is our dominant treatment today is trial and error. Every red dot on the map for venlafaxine or fluoxetine is a non-responder, a known non-responder. Over the course of a year, every blue dot is a positive responder, and the variables separate them, you know, with pretty good coherence. You know, isn't it better if we're doing trial and error to be able to narrow it down and show the error on the report? We don't learn anything from the trial and error clinical trial we're doing abroad, you know, across the world. We do learn something from EEG because machine learning algorithms take every blue dot and every red dot and add to the knowledge, make those discriminant functions more discriminating. And that's really why we've been able to evolve this open clinical registry combined with EEG features into something that is I think a really useful public health utility, addressing a real public health problem. This was created by doctors, it is fed by doctors, and now we've introduced patient-reported outcomes, which is helping us expand that and get more granular. One thing that we learned from working with the military is you get really granular data from patient-reported outcomes, and so that's a place that we're going, that we're not going back. One of my heroes is Tom Insel. Another one of my heroes is Eliot Spitzer. And it's not the scandal you're thinking of. He actually created a scandal, and it was a good scandal. You know, in 2004, he had a litigator who was looking at the drug studies and said, I don't see any of these negative studies being published. And sued GlaxoSmithKline, soon all the others followed, and he sued under consumer fraud statutes. You know, the New York AG said, we have a problem if doctors don't have equal information on both negative and positive trials, and it is a fairness issue to make sure that that information is available to the doctor so they can treat patients correctly. That is the substrate on which we're building. And this New England Journal of Medicine article by Turner came out, said, yes, in fact, we do have a selective publication problem in academic studies and I'll take two examples from the military. There's only 2 on-label meds for PTSD, Paxil and Zoloft. There were, in fact, more negative studies than positive studies, and some of the studies that were submitted to the FDA were, in fact, questionable. They were languaged as positive, but they were really negative or questionable. So we have, in essence, a situation where doctors, through the 90s and the last decade, thought 94% of the studies were positive, when in reality, only 51% of the studies were positive. Kirsch replicated that in his meta-studies by saying maybe there's 1.8% separation from placebo in this body of data. So, you know, our argument is very simple. We're not a perfect discriminant, but if we can do better than 1 or 2%, and if we can do better in an individual patient by reducing trial and error, that is our single simple claim. There's over 100 publications of medication response based on quantitative EEG features. So this is not a new area. In fact, every drug is developed with pharmaco-EEG in phase 1 or phase 2. There are 3 studies, randomized control trials behind peer, and a dozen in the category. And all of our studies have been statistically significant, consistent replication. We've worked with the VA, we've worked with the military, and we've worked with Harvard, Stanford, UCI, et cetera. What's interesting is of these dozen or so trials that are done independently of quantitative EEG features, in fact, many of them have been done in the area of non-pharmacologic remedies, transcranial magnetic stimulation. There are good, strong predictors in quantitative EEG. So we think that's a really fascinating area. I'm gonna skip ahead and be mercifully brief. We're doing 3 replication trials now, one in Southern California with military outside the fence line and veterans and their dependents, one with NATO and the Canadian forces, who are very interested in this and are all transiting the same arc that General Corelli did, another hero, who said, you know, in fact, we've got a much bigger problem than we thought we had. And finally, the Defense Health Agency, which has sponsored our work to date at Walter Reed, and we will continue to move to other locations. So, the methodology, I can review, later with questions, but the basic idea is not to come up with an independent classifier that we make proprietary. We're open science. We take the best of breed, machine learn it, and build it into a tool that doctors can see transparently. And we think that's ultimately the future. In fact, Dr. Insel and I were talking about this. This may be the future of clinical trials. It may be the future of big data helping do what we're unable to do or fund with small data in academic settings. This is ultimately what Sir William Osler said, is every prescribing decision involves a risk-benefit, and we think the best way to position doctors is with some idea about what the true returns are going to be. Thank you.
Thanks, George. I'm sure there'll be lots of questions, and it'll be great to hear what the audience thinks about much of this. Jay Lombard is co-founder and chief scientific officer and medical director at GenoMind. He's responsible for GenoMind's scientific research and development, as well as medical leadership and clinical oversight. He's a board-certified neurologist. He's published several books on the role of nutrition in the brain and has lectured extensively on this topic, has had numerous television and radio appearances, including Larry King, Dr. Oz, CBS, Fox News, and on. Also presented at a TEDMed 2012 lecture, and he's currently professor of neurology at Touro College of Osteopathic Medicine, and I think is going to go without PowerPoint.
That's right.
Terrific.
I'm going naked here.
Yeah. They say that if power corrupts, then PowerPoint corrupts absolutely. So here's your chance.
Kind of reminds me of what was the spaceship that actually crashed, and they were saying how the PowerPoint presentation was part of the reason that they were not able to detect what that issue was. Everyone was kind of distracted by the PowerPoint presentation. So I'm taking their lead. But first of all, it's great to be with another fellow neurologist here, Dr. Gordon, who I've known for a long, long time. And Dr. Insel, I'm going to actually speak a little bit on his behalf because a lot of the technology that Genemind is based upon really came out of the conceptualization of psychiatric disease being not categorical, but dimensional. I want to explain a little bit about what that means. First, an anecdote. I gave grand rounds about a year and a half ago at one of the major teaching hospitals in psychiatry. And the lecture was about using biomarkers to treat mental health disorders, predominantly genetics, but talking also about imaging, EEG technologies. It was a pretty broad-based lecture about all these different tools. And it was a very depressing experience for me because at the end of the day, I left there with a feeling that the only questions that the psychiatrist had for me was, does insurance pay for these things? And that was a very depressing sort of comeback for a lecture I put a lot of time into. And I asked the chairman of psychiatry, I said, why is it that psychiatrists have such little interest in understanding brain science? And he said to me that they didn't sign up to become neuroscientists. And I thought that was really interesting, how we've basically allowed a profession to treat the brain with very rudimentary understanding about the organ. I mean, can you imagine a neurosurgeon going in and dissecting a brain tumor and not knowing the differentiation between, the gray matter and the white matter? I mean, you would go crazy. You would never allow that to happen. But why is it we allow psychiatrists to be able to prescribe medications which alter the brain in structural ways? And yet we just say, okay, I guess your opinion is right. Let me take Lexapro instead of a mood stabilizer. So that was kind of the incentive for developing Genemind. And the basis of our company really is based upon Dr. Insel. So I don't know if you know anything about what we do because we've never really spoken before about it. But essentially, if you look at psychiatric disease as sort of an overarching theme, a 5,000-foot perspective of what psychiatric disease actually is, at least in Jay Lombard's opinion, is that there's an imbalance between emotional centers and rational centers of the brain. So we can go into the anatomy, talk about the frontal lobes and executive brain function, and also talk about limbic lobes and the amygdala and sort of emotional valence of experience. But most psychiatric disease, in one way or the other, are characterized by imbalances between sort of that rational brain and that irrational emotional brain. And we know the neurocircuits that are involved in this. We know a lot about the biochemistry that's involved in this, and we know a lot about the genetics that also relate to these abnormal relationships between this balance of frontal lobe and limbic lobe activity. And that really allows us, whether we're doing genetic biomarkers or EEG biomarkers or imaging biomarkers, to begin mapping out sort of the circuitry, if you will, the visualization of something which is phenomenological. My TED Med talk was about the difficulty, quite frankly, of really creating biomarkers for conditions that are not like neurological disease. A person has a stroke, you do an MRI scan, you can specifically see what area of the brain is affected and if it's caused by an embolus or a thrombus. In psychiatry, these diseases are not localized diseases. We can't pinpoint specifically where in the brain anatomically they are. It doesn't mean that they don't exist. We know that they exist. It's just that it's very difficult to get that kind of specificity. And the reality is that again, this is Jay Lombard's opinion. I'm curious, we'll open this up to my esteemed colleagues here, is that psychiatric disease are really characterized by a sense of disconnectivity. If you look at some newer technologies where we can actually assess connectivity in the brain, we see that the reason we can't localize psychiatric disease is because they're non-localized. They're diffuse across the brain. There are communication hubs or networks in the brain that are not properly communicating with each other that creates these imbalances, whether they're biochemical or anatomical or even environmental. So the goal of all of us in this field is to sort of change the conversation, if you will. Instead of a person having a diagnosis of depression, which is a label. It's a diagnostic categorical label where we're relying on subjective information that a patient's giving us. And think about the inherent room for error when we're asking a patient to report back on their inner subjectivity in situations where the disorder itself is affecting their communication, right? So if you have a geriatric patient who's developing attentional problems and depression, and this is the first sign of Alzheimer's disease as opposed to, some other psychiatric problem, how are we able to elicit as clinicians what's really going on in their brains when they are subjectively reporting something that they themselves may not have even awareness of? Very often as a clinician, the patients with Alzheimer's they never came to my office saying, I have cognitive problems. It was always a spouse or a sibling or a child that would say, hey, Dad, you're forgetting a lot. No, I'm not. Leave me alone. There's nothing wrong with my memory. You're trying to steal my savings or something like that. So there's this inherent blind spot that we have in psychiatry, and yet we're asking patients to report back to make diagnoses. So the purpose of biomarkers is to kind of open up that window to see what the biology is of these conditions, whether it's genetics or EEG or imaging or all of the above, to allow physicians to understand how to be much more specific in their intervention, whether it's video games or it's nutritional factors or it's drug. It's giving us the opportunity to have a much finer tune understanding about biology in psychiatric disease as opposed to guesswork. And that's basically what all of us are trying to do in different ways. I think that there'll be a convergence of these technologies in the next couple of years because they're all important. They all have their limitations as well as their strengths. I'm happy to talk about what the strengths are of genetics in psychiatry because it helps us to pick up higher rates of adverse drug effects for patients. The biggest reason for noncompliance is that people feel worse on medication. They don't feel better. They feel worse. And the typical response that patients get is, hang in there. You'll get better after a couple of months. So I just spoke to an ENT specialist who's depressed and has no libido, is not able to sleep, has panic attacks, and is on an SSRI and is getting worse. And his psychiatrist said to him, look, we'll give you some Klonopin to help you sleep. We'll give you Ambien if that doesn't work. And we'll give you Viagra to help your erectile dysfunction. And for your anxiety, increase your Xanax dose. I mean, this is the reality of what's going on in psychiatry today. This is not make-believe. And think about, from a patient's perspective, how reckless that type of behavior actually is. So our goal is to sort of improve. And it's kind of ironic that two neurologists are here talking about how to improve psychiatry, but it's been led by Dr. Insel, because what Dr. Insel has said is that we need to change this categorical diagnosis away from labeling patients to saying that you are autistic. That means a lot of different things. There's a lot of different types of autisms. There are subtypes that are based upon very different biological factors. If we think it's a one-size-fits-all, we will be the same place 10 years from now that we are actually where we are right now. And we just can't let that happen. We have to change the conversation in a way like Dr. Insel has suggested. And I'm, quite frankly, very honored to be here with you, Dr. Insel. So that's all I have to say.
Thank you.
Take questions. Thank you.
So we want to open this up, but before we do that, just to draw out a couple of points. And what we heard from Eliot was about the use of both cognitive testing, a little bit in genetics, and some of the work with MRI as well as QEEG. George really focused on the power of physiology of QEEG. Jay, do you want to say something about genetics specifically in terms of genome-mind and
Sure.
the sorts of biomarkers that might be on the horizon from genetics?
Absolutely. So going back to this framework of looking at how genes affect behavior, because we're all born with different temperaments. I'll never forget, I was actually on vacation, and I met a child psychiatrist. And he said, what do you do? I said, I'm a co-founder of a company that's looking at genetics and psychiatry. And he says, genetics has nothing to do with psychiatry. And I said, look at my 2 children, okay? They are born from the same parents, I think. I don't know. My older daughter doesn't look that much like me. They are completely different children. I mean, as opposite as night and day in everything about them. And they came from the same genetic pool, we believe. And so to dismiss genetics as not playing a role in psychiatric disease is just to be ill-informed. So what we're looking at, the primary areas that we're looking at are ion channel genes, because ion channels are related, and we'll talk about pharmacokinetic genes as well, but for this audience, I think that would be less interesting, Tom. But the ion channel genes, what I tell psychiatrists is that they need to think of themselves as cardiologists of the brain, because all of us, our brains are in this constant yin-yang, activation, relaxation. And in fact, EEG measures this, overactivity, underactivity. And these circuits are like light switches that go on and off. Imagine a city full of flickering lights that are being activated. That activation and inhibition of the brain is primarily related to ion channels. Okay, ion channels control they're the light switch of the brain, basically. And what's fascinating about these ion channels are that they've been associated with psychiatric disease across diagnostic boundaries. That's what's so interesting. In other words, there's a particular ion channel called a calcium ion channel that has been shown in these large genome-wide association studies, thousands of patients, to be abnormally, to a greater degree, whether you're bipolar, autistic, schizophrenic, PTSD, or depressed. What that tells us is that there is a common substrate for psychiatric disease beyond just a particular chemical or beyond just a particular area of the brain, that there's an imbalance between activation and inhibition of the brain mediated by these ion channels. And in fact, when I tell psychiatrists that they're cardiologists of the brain, when we use mood-stabilizing drugs, whether it's anticonvulsants, fish oil, omega-3 fish oil, Dr. Hiblin's going to speak about lamotrigine or other medications, we are changing ion channel activity. So the excitement that I have personally about these findings is that there's a sort of a final common pathway of convergence of disruption in many psychiatric disease caused by these ion channels and their dysregulation genetically. And what this allows us to do opens up a whole new understanding about prescribing medications that would normally not be considered. We've had many, many patients who had ion channel gene disorders that have migraines, bipolar symptoms, cyclical mood disorders that were not classically diagnosed as being bipolar because they didn't meet the pure criteria of bipolar, who respond phenomenally to a low dose of an anticonvulsant medication because the clinician was able to recognize that the ion channel was the primary culprit, if you will, of that psychiatric disease. So that's probably the major areas in a nutshell that we're excited about. Of course, the pharmacokinetic genes are, looking at drug metabolism. Right. Dr. Gordon mentioned, the reuptake of certain medications, and that's also, I think, very useful for psychiatrists to have as well.
So usually when there's a discussion about biomarkers, there's a set of questions that come up right away, and I like to call them the 3R questions. There's a question about rigor. So is this really ready for prime time? Is it going to be useful not in 1,000 patients, but in a person in my family? Can we reduce this so that it will be practically effective and helpful? So that's the rigor piece. The second is a question about regulation. So we have now FDA involvement not only with approving drugs, but they also have a process called qualification process for biomarkers. And they've laid that out on their website in a very thoughtful way to say, this is what we would expect a biomarker to be able to do. And there's different kinds of biomarkers, some that are used for diagnosis, some for predicting adverse events, a range of different things you could think about. And they laid that out, I think, very nicely. The third R is a question that came up after Adam's talk, which is anybody going to pay for this? How does this get paid for? What's the reimbursement strategy that we should think about? And often, at least for public dollar reimbursement, that depends on the second R. It depends on having met some regulatory qualification before Medicaid and Medicare will say, oh yeah, we'll pay for this. This has come up in a huge way in Alzheimer's disease. So there's been a very public battle around who should pay for the very expensive PET imaging that's done for early diagnosis of Alzheimer's. And the FDA has been involved with that. It goes back to the first R. There's been a whole debate about how predictive are the findings, how specific are they. So there's rigor, there's regulatory, and there's reimbursement all the way through. I'd love to just draw the three of you out on any one of those three in terms of where we're at for the kinds of biomarkers you're looking at. If we were to say most of what we've talked about up here has been around depression and its treatment, are we ready for this? Do we have those three R's really lined up? Evian?
So that's a great summary, Tom. So we deliberately decided to try to do all three together because the gap between, in our academic world, between having a breakthrough and real-world implementation can be as long as 15 years. So we tried to really see if we could shorten that dramatically by doing all three of those things in the same time. So the bottom lines, and just by the way, for a quick correction, I'm not a neurologist. Thanks anyway, Jay. I've got a medical and science background, and most of my time has been spent in applying neuroimaging to psychiatry, but thank you anyway. The three things, Tom, is firstly, the rigor that I want to emphasize is that you can never underestimate the importance of numbers in terms of right. Sample size, and it turned out that standardization was a very helpful advance in getting the statistical power and getting that potential replicability, not just in terms of the replication sample, but across sites. So that's the first thing. The rigor is critical and is hugely sample size dependent. The second thing is the reimbursement was the third. The
regulatory.
The regulatory issue was really important. The fact that you introduced us to the key people in STAR-D, who then, with Steve Koslow, the inaugural head of the Human Brain Project, took us to the FDA. And from day one, before we started, they said, here is how to design your study, was absolutely seminal. Because we frankly would not have designed the study so astutely as having a training set with these very specific statistical ways within the training set of looking at the sort of potential replicability of the statistics that are now used in that regard, and then having an independent, second sort of replication sample, and many, many nuances in the way you deal with statistical vagaries. And the third piece, and most important, as you say, we deal with 5 payers through our use in the for-profit side, obviously not the non-for-profit scientific side. And all the 5 payers that we deal with, and they're all the 5 big ones, say the same thing to us. When it comes to cognition, there's already a reimbursement code, 96120, which the American Psychological Association fought for a decade to get that reimbursement code. The Mental Health Parity Coalition is crucial in ensuring that anything that now is scientifically relevant or equivalent, and making my PhD was in cardiology, in serum lipids in the days, that you look at it, you think there's this huge replication in even cardiology. Actually, you would be surprised. There's a lot of scientific evidence in psychiatry that's equivalent, and the Mental Health Parity Coalition helps that. But the bottom line is there is already a CBT code for cognition online, and that's what all these 100, or many of the 100 of the addiction clinics use, and that's made a huge difference. And those 3 pieces together, we are seeing is accelerating the potential adoption of this on scale.
George, what do you think?
Well, I'm going to go after the rigor question because I'm an MBA, so I'm the least qualified to answer a clinical question. But let me give it a shot. We've got 3 trials going on here. We've got an N of 1, an N of 283. I'm going to pick on Sertraline, and an N of 10,000. In the N of 1, this is what doctors do every day. I've had some good experience with venlafaxine, not so much with duloxetine, and I think you look like a venlafaxine person. Great. You know, and they get more and more patients, and doctors get wiser and more experienced, and at the end of the day, you're still dealing with an N of 1 in terms of a heuristic set of kind of thumbnail sketches of what works and what doesn't. So you go to the rigor of an FDA-approved clinical trial like Zoloft, and you've got an N of 283. Not impressive. And these trials are designed so purely they can be injected directly into the veins. There's, 78% of people are excluded immediately. We're looking for unipolar depression. There are all kinds of reasons why what we study for clinical trials for drug approval isn't what happens in the real world. And I'm not sure that advances the science as much as we have conditioned ourselves to believe it does. The N of 10,000 is, if you look at large numbers, what is it that we can learn from large numbers that informs the other two? It doesn't replace them, but is a complement to the other two. And I think, with computing power today and the ability of as Evian said, standardized instruments, I think we have the ability to do some quite remarkable things. I'll give you one example. There's a drug that keeps coming up in our report, Sertraline, MAO inhibitor. No doctor in his right mind will prescribe an MAO inhibitor if there's a chance you could kill yourself with a cheese pizza or a beer. They react with fermentation products in the bowel, and that's not a good thing. But there's a patch now that avoids the gut completely and can lead to no tyramine crisis, no problem. Yeah. Well, in fact, that's it shows up about 9.5% of the time in our work with the military, and they would use it, and they had some miracles. So here's an example where we've got 30, 40 drugs to choose from. We actually show data about old drugs that can actually be the miracle that a patient who's been through all the other SSRIs and SNRIs, et cetera, are not responding to. To me, that's the power of large numbers. And if you look at the history of public health, we've done as much with removing pump handles and epidemiology as we have, I would submit, with the biology that underlies many of these drugs. If we put them together, I think we have the opportunity to really improve patient health.
Terrific.
Yeah.
Jay, anything to add?
Yeah, I want to actually pile on to what was just said here. I think that we have all been very confused in the general public about what data actually means. And that's a big issue. So let me try to just roll this back a second. I think that in genetics, there is tremendous amounts of data regarding the ability to understand psychiatric disease on a genetic basis. The issue is that it's retrospective data. It's not prospective data. So even though you have thousands of patients that are concluded in a genome-wide association study where you're identifying genes of interest, sort of the needle in the haystack, if you will, the question always is, how do you take that data and show that its clinical utility in a clinical setting? And the difficulty with looking at biomarkers prospectively is that we're arguing that a case that these diseases are heterogeneous and therefore require biomarkers, yet we're going to do a study that excludes all the heterogeneity to validate the biomarker. So think about the catch-22 that's inherent in the difficulty of trying to establish a study for, let's say, major depression, looking at any biomarker. And this goes to what you said. When you do drug studies, you are essentially excluding many patients with comorbidity. And then the issue becomes, how do you extrapolate that data to the real-world setting? So you have drugs that are approved by the FDA for a clinical diagnosis, but it's such a narrow population of patients that have been studied. When in the real world, a patient who's taking SSRIs may have diabetes, hypertension, other medications, comorbidity with ADHD or other symptomatology. So we don't have real-world data from the way that we're designing our studies right now. And that's a big problem. I mean, I'm curious to hear, Dr. Insel, how you would,
So I'll respond to your question, but let's open this up as well. We've got about 5 minutes left for questions from the audience. I want to make sure we hear from all of you as well about what you're thinking about in this whole biomarkers, precision medicine area. Yeah, I think the question that you pose is sort of, how does this become relevant to real world? And in a way, you've already heard that answer here. Evian, and actually George as well, stressed the importance of numbers. And Evian didn't say the entire mantra, which I learned from him 8 years ago, perhaps, which has become, I don't have a tattoo, but if I had a tattoo, this is what it would say, which is, it's not only standardization, but it's integration and sharing, and creating these registries, databases that get you the kind of information that you can make actionable because you've got the numbers. You've got, yes, you have 20,000, but within that 20,000, you have the subgroup of 57 who have actually exactly what it is that the person in front of you is struggling with. So maybe it's the comorbidity, maybe it's the genetics, maybe it's the clinical history. That's the way this will move forward. It's actually worked pretty well in other parts of medicine. Been slow to happen here, and getting the registries, getting the numbers has been a real challenge. It's an irony or a paradox of this whole area that for us to understand the N of 1, we have to have the N of 20,000 or of 100,000 to be able to make sense of it. But that's the world of big data, so that the more that you can collect large numbers, the more you can predict individual variation and understand small numbers as well.
Yeah.
Questions from you? Thoughts, ideas, suggestions?
Good morning. My name is Steve Davis. So I'm a psychiatrist. I can respond to Jay that growing up, seeing about half a dozen of my family members suffer with schizophrenia spectrum disorders, the only reason I became a psychiatrist was because my interest in neuroscience and neuroscience research. This appears to me to be one of the most exciting times in psychiatry, despite the fact that we had a decade of the brain over a decade ago, and we didn't really get too far in that area. It feels like we're starting to make a lot of progress, and in the same way that cardiologists have had lots of tools to examine the heart, both from a macro and from a micro level, we are starting to have these same tools around the brain that we can actually use, which is incredibly exciting. Primary care physicians treat the lion's share, the largest number of people with mental health and substance abuse problems. And I'm thinking about the advances that you all are talking about and how it can be 17 years between when an advance is made and when it actually reaches the clinical bedside. How do we shorten that, both with psychiatrists, but even particularly with primary care providers who are seeing most of these patients? Thank you.
Anybody want to take that on?
So one of the experiences for us is, it's a great question, by the way, is coming back to Henry Harbin's point. The difference between psychiatry and a new drug, for example, and the whole new statistics that the FDA have lined up is to take lots of numbers but be able to make predictions on one. I don't think it's fully realized how well thought through this whole process actually is, as Tom alluded to. And I do have that tattoo on my arm, Tom, that says, it says 10,000 to 1. So you need 10,000 subjects, and it's the real world. This is not data that selects out people. You need real world studies. There was no placebo control in our study. It was about relatively how much better is this than treatment currently, randomly in practice. So in answer to your question, it's happening and it's happening fast, and there's a lot of big data out there. It's just the translational piece to get payers, and Mental Health Coalition is just critical game changer in this, to take specific concrete examples that they'll reimburse. I think it's happening much faster than people think.
So let me just add one thing to that answer, which I largely agree with. I think the place where I might differ is one of the places where the times are a-changing is the way in which patients are being empowered to demand changes in their own care. And we're seeing this across all of medicine. It has been slower to happen in psychiatry. But one of the most exciting things that's happening in the world of rare diseases are the developments of registries, like George was talking about, in which these aren't being used by researchers or providers, they're being used by patients themselves to say, I have figured out something, and now I've got to go explain this to my psychiatrist. It's still a question whether they can change the provider's behavior. But if you talk to the big tech companies, I've done this over the last several months, IBM, Apple, potentially even Google, they've sort of thrown up their hands saying changing provider behavior is really hard, but getting patients informed and getting patients to demand better care as a consumer movement, that might work. So that's one of the things to think about as we hear about this, the big data approach or the registries that have developed. Please, last question.
I don't know that there's a straightforward answer to my question, and maybe I'm wanting more to make a statement. I've learned to collect QEEG. I'm submitting CPT codes and getting back rejections. I don't know how to read a QEEG. I rely on a psychologist to read it for me and then to help me figure out how to train the patient. And it seems like, in addition to physicians just not having time to talk to each other and learn from each other, we're spending so much time just trying to get payment. I have trouble just finding out, well, how do I have to code this? It seems like we need help at that level of just making it practical in the office. I'm a part-time practitioner, and it's very difficult, you know. So I know the full-time practitioners, they're not even gonna, it feels like they don't even want to talk about it. I call neurologists to talk about the Q, and they're like, oh, those don't really have, you know, they act like they're silly. And so it's so challenging at just the practical level. I hear you guys are just, I'm so inspired, and I'm thinking, well, tell me right now, what's that CPT code? You know?
Don't go away, don't go away. So before we take an answer to that, can you say a little bit about who actually does the study and how you use it?
I collect the QEEG. I've only been doing it for about 6 months, and now I have a tech helping me with that because it's very time consuming. I send it off to a neuropsychologist who is an expert in reading QEEG and doing neurofeedback training. And I sit with him for an hour. Right now, I'm not charging anybody for that hour. I consider it continuing education. And he describes it to me and tells me how to train. We do 10 sessions or 15 sessions, re-Q the patient. I see the results, and he's helping me learn how to collect brief psych testing, cognitive testing to help have an objective measure. So am I answering your question?
Okay, yeah, George, you want to?
I'm going to give you a straight answer, and I may be wrong, but just in terms of our experience, we found a huge improvement in the amount of EEG billings that are paid for. And that's for 95816 and 95957. So it's the production or recording of an EEG and the interpretation of a digital quantitative EEG, respectively.
Do you find that the interpretation has to be done by a neurologist for it to be reimbursed?
It has to be done by a physician, an experienced physician, but not necessarily a neurologist.
Do they have to be QEEG certified?
No. The tech does. The tech has to be a EEG
Not for the
tech,
collection, for the analysis.
Right, but for the analysis, no. We've had a variety of different folks, psychiatrists, the most advanced technology they had in their office before the EEG was an answering machine.
Wait a minute. Some of them had smartphones.
That's great. Well said.
So before we come to a close here, just a couple of comments reflecting back about what we've heard. I think this has been a great discussion. This is a field that's still emerging. So biomarkers have become integral to cardiology, neurology, almost every other area of medicine. In psychiatry, this is a happening thing. It's not fully baked, but we're on the way. I think there are two things to take away from the discussion. One is that at the end of the day, there's probably not going to be a single magic biomarker, right? It's not going to be that you'll have a blood chemistry number, like your lipid number, that will tell you. And even in cardiology, increasingly what we do is we combine measures from lots of different places. We use imaging, we use plasma lipids, we use clinical history and family history, and now we're beginning to use genetics as well to give patients the best sense of what their risk is for both response to a treatment and actually ultimate prognosis. The second issue to think about is that as we go forward, that mix of tests that we begin to put together are going to be everything you've heard here and probably more. So there will be EEG measures, there will be imaging measures, there will be genetics. There will be some things that you heard a little bit about from Adam, which is using technology and wearable technology, for instance, or something that comes out of measures that you can get off your smartphone. We call these now e-markers. That's kind of the cool new name, not biomarkers, but e-markers. And then what you heard a little bit about from Evian, that wasn't time to go into it, is the power of some of the new cognitive science measures, which are not high-tech, but may be highly, highly predictive, particularly in the earliest phases of psychosis and for relapse for depression, areas where there's such a huge clinical need. So the future is going to be multiplexed. It'll be lots of different tests that will come together much the way they have in heart disease to give us a much better understanding of risk. I think what you've gotten here is a pretty good snapshot of where we are in 2015. It'd be great to do this again in 2017 or 2019. I think you'll see by that time we're even further along on this highway. Thanks to all of you for your interest, and we'll look forward. We're going to have a break at this point, but we'll look forward to discussion.